Hello and welcome back to the Group 3 News. So, our story today is about characterization of putative pathogenic Shewanella algae isolated from ballast water. Without further ado, let's get started. Our group consists of 5 people, which is me, myself, Nur Farisha Izati, Lashini, Marga Arif Afrizal, Nurul Nabila, and also Shahira Tasha. There are 2 objectives for this topic, and the first one is to isolate Shewanella algae from ballast water of ships berthed at Port Kelang, Malaysia, and to characterize Shewanella algae from ballast water of ships berthed at Port Kelang, Malaysia. So, the problem we identify for this topic is, generally, ships carry ballast water to control their stability and trim at the start of the voyage. However, the discharge of ballast water near ports risks the transport of invasive species, including pathogenic species, from one continent to another. It may lead to the emergence of new virulent strains in the existing population. The global distribution of pathogenic bacteria implicates a negative effect on the existing ecosystem as well as marine animal and human health. The physiological diversity and broad respiratory versatility of Shewanella species make it a highly adaptable organism that can survive in different environmental niches.
Next is material and method. To make blood agar, we receive sheep blood from UKM Animal House and human blood from healthy volunteers. For water sample collection, first is ballast water from 9 ballast tanks at Port Kelang, which later fill into 1 liter sterilized shot bottle. Second is seawater at 4 sampling points surrounding port water using Niskin water sampler. For method, we need to isolate and select Shewanella sp from the water sample first. The water sample was serially diluted by stirring water. Then, 0.1 ml of the diluted sample were inoculate on marine agar by spread plate technique. We incubate at 30 Celsius for 3 days. Key characteristic of Shewanella test need to do, and the colony that fit the description was subjected to further test. Next step is to test the biochemical and enzymatic activities. The test is biochemical test, enzymatic activity, and hemolytic activity test. All the tests will incubate at 30 Celsius for 72 hours. Total genomic DNA of S. algae was extracted using CTAB/sodium chloride method. First, the 16SR RNA was amplified using 2 primer. Next, 25 microliter PCR mixture was prepared with a specific PCR condition. For amplification of gyrase gene, we used 2 primers with a condition. The sequencing of the 16SR RNA and gyrase B amplicons were completed by apical scientifics in the heart. The obtained sequence data were aligned and analyzed using bioedit. The species identification was performed by BLAST. For phylogenetic analysis, we used BLAST, Mega version 6.0, and Bootstrap analysis. The biofilm formation by the isolates were quantified based on OD reading, marine broth added for negative control, and suspension of Staphylococcus aureus added as positive control in the assay. Then, we compare the OD with the ODC. Finally, we need to test antibiotic susceptibility using broad-spectrum antibiotics. The MAR index calculated. If a MAR index is more than 0.2, that indicates a high risk source of contamination.
Result for the characterization and identification of S. algae shows that 4 isolates were presumed as Shewanella species from ballast water and 2 from seawater. All these presumptive isolates are gram-negative rod and show circular orange colonies on marine agar. Comparing the biochemical and enzymatic profile of this isolate, all strains are known as S. algae. This is supported with 16S rRNA and gyrB gene sequencing, in nucleotide blast showing more than 99% identity score. For the phylogenetic analysis, there is a bit different for the 16S rRNA and gyrB gene sequencing. The 16S rRNA shows that isolates form a cluster with other Shewanella species. However, the gyrB gene shows that isolates cluster monophyletically apart from the other S. algae. For the biofilm formation capabilities, all strains were observed to be strongly adherent. Next, the antibiotic susceptibility shows that isolates resistance to many types of antibiotic but only sensitive to ciprofloxacin, chloramphenicol, and gentamicin. With MAR index more than 0.2, conclude that isolates were originated from a source that has antibiotic contamination.
Hi, I'm Shewanella and I would like to talk about the discussion. From the results obtained in this study, we can be discussed about the characterization and identification of 4 Shewanella algae isolates. These isolates had key phenotypical characteristics of oxidase positive and sulfide production. Shewanella algae can be differentiated from other species based on the phenotypic characteristics such as the ability to grow at 42 degrees Celsius, tolerance to high salt concentration, and the hemolysis on shipwreck algae. These characteristics group the Shewanella algae isolates into mesophilic and halophilic Shewanella strains. Shewanella algae is confirmed by 16S ribosomal RNA and GRB gene sequencing. Based on the phylogenetic analysis, the GRB gene is more reliable and useful than 16S ribosomal RNA for describing phylogenetic relationship. The isolates show independent branching from Shewanella halihertis and Shewanella hoopney with the similarities values which is lower than 16S ribosomal RNA. The rate of molecular evolution of GRB sequencer is faster than 16S ribosomal RNA, which provides higher phylogenetic resolution. GRB gene has been used as discriminative detection for Shewanella species identification. The capability of all Shewanella algae isolates in this study produce extracellular enzymes such as lipase, protease, DNase, and hemolysin, which could be inferred as part of adaptive mechanism for the species to survive in the marine environment. These could be reducing surrounding microbial competition or degrading organic matter to gain nutrients. However, the adaptation allows for colonization of living hosts if the bacteria enter the host. Next, Shewanella algae was observed to be more virulent species by producing hemolytic activity as a virulence factor. Shewanella algae has an ability to form a strong biofilm formation, which is a significant advantage for the survival adaptation of the species in the ballast tank. The production of exopolysaccharides provide protection from mechanical or chemical treatment and from predatory protease. The biofilm formation may also promote phenotypic modification where genetic exchanges among the microorganisms occur. As for the last capability, Shewanella algae is resistant to antibiotics as encourage host pathogenesis, allowing persistent or chronic diseases. The most commonly antibiotic resistant is against beta-lactams such as amoxicillin, ampicillin, and penicillin, and against cephalosporins such as cephalothene, cefazolin, cefotaxime, and colistin. I would like to conclude that these characteristics may represent the putative pathogenic factor of the Shewanella strains from this study. Thank you.